So last week we stopped at thermodynamics when talking about metabolism. Thermodynamics is the study of energy and energy transfer through physical matter. And we talked a little bit about this when we started talking about our open systems and closed systems. A system is matter and its environment. The outside system are your surroundings. So an open system is a system in which energy is transferred between a system and its surroundings. So a stovetop that is turned on will be transmitting heat into the environment, so it's transferring energy. Biological organisms also transfer energy between their systems and their surroundings. Closed systems cannot transfer energy into their surroundings. So think of things like a pressure cooker. Once that pressure cooker is opened, it will become an open system. The first law of thermodynamics states that the total amount of energy in the system in the universe is constant. Energy cannot be created or destroyed, so there always has been and there always will be the same amount of energy in the universe. Energy can be transferred between things, but never destroyed. So light bulbs can transmit electrical energy into light energy, and so on. The second law of thermodynamics is that every energy transfer increases the universe's entropy. Entropy is the amount of randomness or disorder in a system. So the more entropy there is, the more disorder and the less energy. Some energy in reactions is lost and unusable for purposes at hand, as heat energy. So the more energy that a system loses to its surroundings, the less ordered and the more random the system. So every time there's an energy transfer that occurs, the more entropy occurs. So those energy transfers that we talked about for the first law aren't as simple as they might seem just by stating these transfers of energy. We have a lot of entropy that occurs because of these energy transfers, and some energy loss is heat energy, which is unusable for the purposes at hand. So living things are in this constant uphill battle against universal entropy, trying to keep things in a sense of order in a universe that is constantly disordered. Adenosine triphosphate, or ATP, is the cell's primary energy currency, and we've talked about this before. So ATP is adenosine with 3 phosphate groups. The bonds that link these phosphate groups are phosphoanhydride bonds. They are very high energy, so when they break through hydrolysis to release energy from ATP to ADP, that energy can be harnessed in chemical processes. It's a highly unstable molecule, ATP. It can spontaneously dissociate. So energy that is used by one reaction is used to drive another reaction. It's called energy coupling. You may remember this as secondary active transport. So it's along for the ride in a process that uses a lot of energy, or ATP. So the sodium-potassium ATPase pump is a primary active transport system. So a lot of systems use the energy created through the sodium-potassium pump as an energy couple for their physiological processes. Phosphorylation is the chemical addition of a phosphate group to a compound. In ATP reactions, that free-floating phosphate group can actually be used in energy coupling reactions. So we've talked a little bit about enzymes in the past. Remember, enzymes are a type of protein. They're very specific. They catalyze or speed up chemical reactions. They lower activation energy. So remember, activation energy is the amount of energy needed in order for a chemical reaction to occur. So by enzymes lowering activation energy, they are speeding up chemical reactions. They're allowing them to happen with a lower amount of energy. Most enzymes will end in ACE. They do not change the delta G, or the change in Gibbs free energy. And they do not change whether or not a reaction is endergonic or exergonic. They simply speed up a reaction. So substrates are chemical reactants that an enzyme binds to, or rather, that binds to an enzyme. The active site is the location within the enzyme where the substrate binds. This will be very substrate specific. They will have an exact shape that will fit the substrate that the enzyme reacts on. We've talked in the past about optimal conditions for enzymes. So we know that different temperature and pH changes can influence the binding of these substrates to these enzymes. So high temperatures or unsuitable pHs for enzymes can denature the enzyme, which would not allow that substrate to bind to that enzyme. In the past, the model that was used the most in talking about this enzyme and substrate complex was the lock and key model. The lock and key model states that enzymes and substrates bind together perfectly and typically within one step. Now, as time has gone on, scientists have noted that it's not as simple as just this one-step process. So now we use a model called induced fit. So it's a more dynamic reaction between this enzyme and substrate. There's an intermediate step, which is the formation of this enzyme-substrate complex. During this enzyme-substrate complex, it's very unstable and reactive, so it will want to return to its original state after this reaction. The enzyme will never be used up. It will always return to its original state. So as we can see here, this enzyme has an active site that is specific to the substrate. When the substrate binds, it forms an enzyme-substrate complex. The enzyme works on the reactant to form a product, and the product leaves the active site of the enzyme. The enzyme is unchanged. So the control of metabolism depends on the cell and the location within the body's tissues. So the cellular needs vary from cell to cell. Metabolic needs vary among fat storage cells, skin cells, nerve cells, etc., or different types of cells within plants or different animals. So the energetic and enzymatic needs vary depending on the location. Rates of reactions will be determined by the efficiency of enzymes. Are they within their ideal temperature, pH, and simply conditions? If these enzymes are within their ideal conditions, then the rate of reactions should speed up. Enzyme activity can also be promoted or inhibited. So the three types of inhibition that we will be talking about are competitive inhibition, non-competitive inhibition, and allosteric inhibition. Competitive inhibition is when an inhibitor molecule looks similar enough to the substrate that it can bind to the active site of the enzyme. So it will bind in place of the substrate, inhibiting that enzyme from working on its specific substrate. In non-competitive inhibition, an inhibitor molecule will bind to the enzyme at an allosteric site. So this is a site that's not the active site, and it will block the substrate from binding. In allosteric inhibition, again, we have a molecule attaching to the allosteric site, not the active site, but this time it's going to induce a conformational change within the enzyme so that it has a decreased affinity for the substrate. Maybe the substrate won't be able to fit in the active site anymore. On the other hand, we also have allosteric activators, so they do the opposite thing as allosteric inhibitors. So when these molecules attach to the allosteric site, they actually induce a conformational change that will increase the affinity for the enzyme for the substrate. We also have cofactors and coenzymes. These are going to promote optimal conformation and enzymatic function. So sometimes enzymes won't even be able to function without their cofactors and coenzymes. Sometimes they are bonded to these enzymes via covalent bonds, and they can't function without them. When we go through the next chapter and talk about different aspects of cellular respiration, you will be able to see these specific cofactors and coenzymes that are working toward our most important chemical processes. Cofactors are inorganic ions, so these are going to be things like minerals, microminerals that you get from diet. Coenzymes are organic helper molecules. These will be things more like vitamins. Enzymes are compartmentalized into organelles, and this allows them to be more efficient for their chemical processes that they perform and catalyze. So enzymes involved in cellular respiration will be housed in mitochondria, where these where cellular respiration takes place. Cells have also evolved to use chemical reaction products to regulate enzymatic activity. So think of this kind of like negative feedback. So if you have an abundance of products, it will slow down the reaction so that more products will not be created in surplus. And this will save some energy in the organism.